All right, guys, we are going to get into DNA replication. Now, before we jump into DNA replication, I want us to take a moment to have a quick review. So, I want you to sit and be able to answer and recall all of the following questions. All right, very, very good. Now, DNA replication. Whenever cells divide through the process of mitosis, DNA must be copied. So, DNA replication is just the process of making a copy of DNA. The DNA made during DNA replication is going to be an exact copy of the original DNA strand. Now, remember, this happens in S phase of interphase. Now, just a quick recap in case you can't remember. The bases in DNA have to match up in a certain way. A is always going to pair with T, while C is always going to pair with G. Therefore, DNA can be used as a template to make new DNA. An example of this would be if you know one side, you are definitely going to know the other as long as it's following those rules. So I have an example sequence down here. On the top side, you've got AG, TCCA, and on the bottom, you see it is perfectly matched and paired. It is following those complementary base paired rules. Since DNA replication uses existing DNA as a template, it is considered semi-conservative. What we mean when we say semi-conservative is that it means that each DNA molecule consists of an original strand and a new strand. So as you can see our little DNA cartoon over here, they are very happy about being a semi-conservative molecule. So with this kind of model, you can see represented by the different colors that we have our two different strands. Our original strand is going to be represented by our blue line over here, while we have our new strand that is coming in green to wrap around it to make our two exact copies of this one DNA molecule. So DNA strands are going to be held together by hydrogen bonds, but those hydrogen bonds are weak. This is what makes DNA so easy to replicate. So this is where hydrogen bonds are going to be found. This is guanine and this is cytosine, and the bonds are held here. So copying DNA requires many enzymes, which we recall are proteins, but we're going to be focusing on these three specifically. First, you have helicase. Helicase is the enzyme that separates DNA strands. Next, you have DNA polymerase. DNA polymerase adds nucleotides and it proofreads the sequence to make sure that no errors have occurred in the process. And finally, we have the ligase enzyme. This is what links our new pieces of DNA together. Now, the enzyme helicase unzips or it separates the two strands of DNA. Like when you're unzipping your jacket at the end of the day, you are bringing the zipper down to make two separate parts instead of having one united zipper. That's how helicase works. It is unzipping those two strands of DNA. Now, your DNA polymerase adds the nucleotides of the new strands using the old strand as a template, which I have some pictures that'll help make a little bit more sense. I know that's a little hard to read and understand. And finally, you have ligase, which is what is going to link together the pieces of the new DNA. So think of your ligase as the new zipper you put on that's going to put our two new DNA strands together. So here we have an excellent diagram to help show that. So here, we see the helicase, which I told you is the enzyme that is unzipping. Because this side used to look like this side, but the helicase is coming in and unzipping it. So that way, over here, we're able to have two separate strands. So here's your DNA polymerase. And if you recall on our last slide, the DNA polymerase is what adds the nucleotides to the new strand using the old strand as a template. So if you notice, this is where we are starting to build it. We have our free nucleotides that are coming in and adding itself on to our DNA template. Now down here we have our ligase. Our ligase is acting as a zipper to help these things come together to create two new strands of DNA that are exactly like the strand that we started with. So first, we are going to start with our helicase. The second step is going to be the DNA polymerase, adding our new nucleotides in to help rebuild our strands. And finally, we're going to have our ligase that's helping everything be put together once the DNA polymerase has added the nitrogenous bases to our strands. So, let's do some fact checks on replication. It takes cells around 6 to 8 hours to replicate your DNA. That's insane. Your cells have replicated trillions of times in your lifetime alone. So imagine thinking about all the DNA replication that has happened all over the world. How many times that would be? What an amazing number. But mistakes do happen sometimes. But DNA polymerase fixes it most of the time. Now, we'll talk about what happens when mistakes don't get fixed a little bit later. But just like you and I, DNA replication is not perfect, and mistakes can happen, and they're not always corrected. Errors are normally limited to around 1 per 1 billion nucleotides. So that is a very, very small chance for a mistake.